All right. Welcome, everybody, to Standing for Truth. I think we're going to have some fun with this specific video. What are we doing in this video exactly? Well, we are demolishing Paulogia on the so-called best evidence for evolution. Now, I might break this up into two separate parts because two of what he believes are the best lines of evidence for evolution include endogenous retroviruses, which I've written an entire book on. I've engaged in many formal and informal debates on. I've also given a series of lectures. I've put out videos. I've refuted the critics. And so I'll be having a lot of fun with that one. And that alone might take a couple hours. As everybody knows, I like to be comprehensive. His other line of evidence, one that according to him, which tells me he didn't look into it too good, is one that was a major line of evidence apparently that helped him convert to believing that whales, pine trees, humans, and banana plants are related through common ancestry, was the human chromosome 2 fusion. And I know you're thinking, hasn't that already been overturned? And yes, it has. And so we just did, Matt and myself, we did a video couple nights ago debunking Gutsick Gibbon, really just refuting all of her arguments. She put out a challenge, absolutely decimated it. It'll be really interesting to see how Gutsick Gibbon gets out of this one. I don't think she'll be able to. And so if Gutsick Gibbon can't get out of it, if Gutsick Gibbon can't answer our challenges here, and definitely Paulogia, somebody who relies on people like Erica, Dodgeball, Dan Stern, Cardinal, people like Paulogia, these content creators on YouTube that promote the idea that they're related to strawberries, essentially. They look to people like, again, Gutsick Gibbon and the experts who we actually refute with sophisticated, very technical arguments. And therefore, if again, people like Erica and Dan can't answer our challenges or counter us, then of course, people like Paulogia, who are essentially just repeating arguments from these people, they're not doing any original research or coming up with their own arguments, which is why you'll never see them in the hours and hours worth of open mic debates that we do. For example, last week we had it went nearly 8 hours. No, I think it actually went 8 hours. And I engaged at least two evolutionists, frequent debaters in the community on the chromosome 2. So you'll never see people like Paulogia engaging in the debate dojo on these topics. I've also engaged Dr. Dan Stern Cardinal on the fusion. He failed to address the major challenges to that argument. And so if you want to see all of those exchanges, debates, discussions, either check the description box or check the channel and the website, standfortruthministries.com, for endless content. So yes, debunked. The other day, Matt and I went 3 hours. So this won't be quite as comprehensive as that one. We're going to have some fun here. But if you want to see more on the chromosome 2 fusion, do check out this video. Also, I want to thank everybody for helping us hit 3 million views. Our supporters, our listeners. Again, this is a listener supported ministry, essentially. You know, I'm doing this full time, which is why I'm able to put on 4 to 5 shows a week, which is why we're able to host so many events on different topics. We host interviews, discussions. Once a year, we put on our Defending Genesis conference. We've published now about 16 books. And we put out a lot of articles. So we're doing a lot here, guys. I've got a podcast with now approximately 140 episodes uploaded. And so God bless you. Praise the Lord. You know, we got the best audience on YouTube. I appreciate how engaged everybody is, especially in the live chats. And this specific video is pre-recorded. But as people know, for the most part, we do our shows live. And we have a lot of fun with them, too. We also have our secondary channel, Young Earth Creation. And so between that channel and this channel, I think we're at about between 43 and 45,000 subscribers. So again, I want to thank all of our supporters, all of our listeners. You guys are awesome. So with that, let's get right into it. So I am going to share screen, and I am going to make sure that I've got the audio. Here we go. Share system audio. So typically I've got my awesome audience in the live chat letting me know if audio video is coming through. So I've got the system audio shared. Let's share the screen. What I'll do is play, excuse me, I'll play Paulogia's video a little bit, maybe 30 seconds here, 30 seconds there, and then I'll respond. Right off the bat, in a nutshell, I can tell you that the evidence for the so-called human chromosome 2, it's incredibly flimsy. Paulogia just really goes into like the basics of it, doesn't try to refute any of our counters. But firstly, there was no fusion, the so-called fusion site. It's too degenerate. It's only about 798 base pairs. Healthy telomere sequences range between 5,000 to 15,000 bases in humans. The telomeres are the end caps of chromosomes. And so this specific DNA sequence therefore exists at the end of chromosomes. And again, they range from 5,000 to 15,000 bases. And so you're looking, if there was a fusion at a signature, between 10,000 to 30,000 bases long. Even if it degenerated significantly over time, you're still looking at a signature in the thousands at least. And all we get is a pathetic 798 base pairs. Not to mention the fact that the entire area is actually overlapped by a highly functional and essential gene, the DDX11L2 gene. And the fusion site itself is an active promoter. There's 2 promoters in it. Promoters are basically genetic switches. They're important to the genome and to the function of biological organisms. And so this is an active promoter for a highly functional gene. Paulogia answer this question. How do you get highly functional genes? By slamming together 2 chromosomes. I want something better than the usual story that I'm provided in debates, because stories are not empirical science. Okay, these types of fancy stories that we get to our questions, such as this question here on the DDX11L2 gene that basically spans the so-called fusion site. They're not science. You can believe in them, but they're not science. They don't belong in science textbooks. The so-called telomeric repeats, TTA, GGG, CCC, TAA, that's the reverse complement. DNA is double-stranded, as everybody knows. Those are found as interstitial repeats in all throughout the genome, in all chromosomes. We're going to go over all of this in detail, just kind of giving you the chromosome 2 beatdown in a nutshell. Paulogia, if you disagree, let's debate the chromosome 2. Let's go. I'm ready. These sequences, these interstitial telomeric sequences, significant activity has been detected in them, at least in one secular paper. And then also Dr. Jeffrey Tompkins, PhD geneticist, he has done significant work looking at these sequences spread out across the entire genome in all chromosomes, not just at the ends in the telomere regions, but all throughout the chromosomes. And they have nothing to do with fusions. They function in gene expression. And your more informed evolutionist like Gutsick Gibbon, she would say, well, okay, yes, this is the case. You got us on this one. But the fusion site, the so-called fusion site, I should say, is the only place that has the sequences in both forwards and in reverse, essentially in a head-to-head organization, TTA, GGG, CCC, TAA. That is false. That is erroneous. It's a bad argument. Doesn't work. Obviously, critics who use that argument haven't looked in the genome. My brother in the Lord, Christopher Roop, and I, we've written an article together, Genetic Entropy, Refuting the Critics. And we've done some work on this. I recently put out an article on it where we discovered the sequences in both forwards and in reverse in multiple chromosomes with a very strong signature, a very pristine signature in chromosome 9, which we're going to touch on as well. The so-called cryptic centromere, the second deactivated centromere that should be found, has not been found. what they do point to, these alphoid repeats, again, those are found scattered throughout the genome, and the signature there is not what should be seen or expected if this really was the remnants of a second centromere. And it turns out that region is also overlapped by a functional, an essential gene. And so what we see in terms of the so-called signatures that people like Paulogia look to as evidence for chromosome 2 fusion, they actually represent functional components inside essential genes. And that's it. Game over for the chromosome 2. With that being said, let's see what Paulogia has to say, and let's see if Mr. Paulogia here has actually addressed any of the points that I brought up, or is he basically just repeating arguments that we've already refuted over and over again?
We don't deny the measurements taken from a footprint just because we didn't watch it being made or can't make another one just like it. No. What science cares about most is predicting future data. The better a model makes predictions of findings, the better it is. Be it the timing of the next eclipse, the existence of the Higgs boson particle, or genetic markers that make sense only if all life emerged from common ancestry. One such prediction is the fusion of human chromosome 2. This one was so definitive for me that I named my pre-Paulogia blog Chromosome 2. In a live debate, I'd obviously go over this myself, but since this is a video, let me throw to the Nova documentary on the Kitzmiller versus Dover trial. The legal case.
Notice how he says this was just definitive proof. And I can predict that he knew nothing about the functional nature of the DDX11L2 gene. He knew nothing about these interstitial or internal telomeric repeats that are found scattered all throughout the genome. I can guarantee that he knew nothing about the fact that we actually find these sequences in forwards and in reverse in the head-to-head organization in other chromosomes, specifically chromosome 9. I can guarantee he had no idea that these interstitial telomeric repeats that have nothing to do with chromosome fusions scattered throughout the genome are functional in gene expression. I would also predict that he had no idea that the site was so degenerate, and that the authors of a prominent paper on the chromosome 2 fusion actually asked the question, why is this area so degenerate? And they follow up that question because they were surprised. This was unexpected. They did not predict the degenerate nature of this site. They actually followed up with at least 3 possible reasons why it's so degenerate, and all 3 reasons are incredibly weak, unimpressive, and not convincing. And I bet Paulogia could not reiterate those reasons, because evolutionists like Paulogia, they are just looking for confirmation bias. They so badly want to believe that they're related to strawberries and whales, and ultimately go back to a single-celled-like ancestor millions of years ago, billions of years ago, that essentially evolved from non-living chemicals in some nice warm pond. They so badly want to believe that, rather than believe in the God of the Bible, who spoke the universe into existence. In the beginning, God created the heaven and the earth. And so it's kind of a shame that this, to him, was so definitive, but he was not. And again, I predict, he can correct me if I'm wrong, but I'm pretty sure I'm right here. He was not aware of all of these various points that I am mentioning here. Okay, let's continue.
Where intelligent design was ruled to not be science.
The cells of all great apes, like chimpanzees, gorillas, and orangutans, contain 24 pairs of chromosomes. If humans share a common ancestor with apes, you'd expect us to have the same number. But surprisingly, human cells contain only 23 pairs.
Evolution makes a testable prediction. And that is that somewhere in the human genome, we ought to be able to find a piece of Scotch tape holding 2 chromosomes together, so that our 24 pairs, 2 of them were pasted together to form just 23. And if we can't find that, then the hypothesis of common ancestry is wrong and evolution is mistaken.
Typically.
Notice that. He said, if we can't find this fusion, then the hypothesis of common ancestry is wrong. In other words, if there's no fusion, there's no evolution. Imagine putting all of your hopes and dreams in one single pathetic argument. Because yes, we as humans, we have 46 chromosomes, and then your great apes have 48 chromosomes. And to explain away this discrepancy, they'll say, well, 2 ape-like chromosomes in the human line after the split, right? They say humans and chimpanzees go back to a common ancestor about 6 million years ago. And sometimes after that divergence, where the human line went one way and your chimpanzee line went the other way, there was a supposed fusion in the human line between two chimp-like chromosomes, which is actually have been renamed in the chimpanzee to 2A and 2B. That's how sure that this so-called chromosome fusion occurred. And let's continue, but I just wanted to point out, yes, if there's no fusion, there's no evolution. And the fact that these evolutionists can't actually address our challenges and our arguments tells us that the evolutionary story is pretty weak right now.
On the ends of every chromosome, you should find special genetic markers, or sequences of DNA, called telomeres. And in their middles, you should find different genetic markers called centromeres. But if a mutation occurred in the past, causing two pairs of chromosomes to fuse, we should find evidence in those genetic markers. Telomeres not only at the ends of the new chromosome, but also at their middles, and not one, but two centromeres.
Lo and behold, the answer is in chromosome number 2. All of the marks of the fusion of those chromosomes predicted by common descent and evolution, all those marks are present on human chromosome number 2. So the case is closed in a most beautiful way. Evolution has made a testable prediction, and it has passed.
Once again, on the few occasions where Kent has even attempted to address human chromosome 2, he has missed the point entirely.
Help me whack an atheist. Chromosome.
So the question is, have they actually found these signatures? Well, again, Paulogia here is taking the lazy way out. He's just playing somebody else's video, this popular video, just repeating the very basic arguments for the so-called chromosome 2 fusion. But as you'll see, he doesn't attempt to address any of the major responses to those arguments, nor does he have any of his own original research. So since this video is obviously going to be limited, I put out an article here, comprehensive, about 16 pages, overturning the human chromosome 2 fusion. And so Dr. Ken Miller, who was very prominent in essentially promoting this argument, I refute him in this article, as you can see here, Debunking Ken Miller, and I go through all the various counter rebuttals to basically the points that I've made and what I just made in my nutshell explanation, leaving no stone unturned. This one right here is my recent article to Gutsick Gibbon and refuting her video titled Donny Botches Human Chromosome 2 Again. I'll touch on the data in this in a little bit, but not until we reach that portion of this program. So let me go over a couple slides here, reiterating a few points that were just brought up, and also demonstrating why the so-called signatures that the speaker here says are found are actually not found. Let's go full screen so you can see it better. Okay, so as you can see here, this is a depiction of the hypothetical chromosome fusion. You got ape-like to chimp-like chromosomes here, 2A and 2B. Again, you've got your telomeres. They're the end caps of chromosomes. So if you had two chromosomes slammed together, right in the middle, you should have the evidence for this unilateral mistake in the center with all the telomeric repeats. And so hypothetically, they fuse together. You'd have your telomere DNA here. You'd have your one centromere here, and then your second centromere. Okay. Here's the chromosome number 2. So this is the prediction that has been made. Actual telomere on the chromosome, so you still have your end caps, which actually telomeres, one of their jobs is to protect from fusions. You can picture your telomeres basically like your shoelaces. At the end of your shoelaces, you've got your piece of plastic that protects the shoelaces from basically coming apart and breaking down. But as that plastic piece breaks or tears off, then your shoelace starts to flare out and break down. So that's why these telomeres are so important, is because they're protecting the chromosome. So here you should have an active centromere and then an inactive centromere with your telomere DNA sequences in the middle, found in the middle. But again, I pointed out earlier that this telomeric DNA are approximately in humans, healthy telomeres, between 5,000 and 15,000 bases long, which means you should have a mark or a DNA signature, if this were really the ancient remnants of a fusion, of between 10,000 and 30,000 bases in length. And so the evolutionists have to either say, well, in order for the fusion to occur in the first place, the area must have degenerated before fusion took place, and so the area would start off degenerate and small, or over the last 4 to 6 million years, whenever the fusion would have taken place in the hominin line, in the human line. Firstly, genetic fixation is slow, and whatever individual gets this fusion, eventually this fusion is going to have to take over the population, since every single human being today, guess what, has 46 chromosomes. Where's the subpopulations on the planet today that have the 48 chromosomes? And so I've heard some evolutionary scientists like Dr. Joel Duff say, well, at the time you'd have the fusion spread throughout a subpopulation, but then all your other populations around the globe, they'd go extinct, and then all that's left is basically the 46 chromosomes, the humans with 46 chromosomes. But a lot of this, especially coalescence, we talk a lot about coalescence here, depends on random mating. But there's not random mating, because historically you have tribes, you have people isolated in tribes, in groups, and typically they would marry within those groups. And so you'd have tribes spread out around the globe. And so it's pretty difficult to have a fusion, 46 chromosomes, spread to everybody, because again, you're going to have to invent fanciful storytelling on how this fusion did spread in the first place. But anyways, this would have had to occur between 4 and 6 million years ago, sometime after the split. And according to them, the area should have degenerated significantly to the 798 base pairs. But it's funny. So given what, another 100,000 years, 500,000 years, a million years, according to the evolutionary model, apparently, if it's this rapidly mutating and changing, and it's a spot in the genome that's basically a hot spot for degeneration, then the evolutionists are going to have to tell you, well, in 500,000 to a million years, this site is going to, there's going to be nothing there to see. It's just going to degenerate to nothingness. So there's a lot of interesting storytelling going on. And basically, here's the prediction. His video that he played, the speaker is trying to say that this prediction was met. But the fact is, it was not met. But anyways, the earliest hint at a chromosome fusion in the human line, right after this hypothetical chimp human split, was actually prior to genetic sequencing. And scientists were looking at images of human and chimpanzee chromosomes through a sophisticated technique. It's a very impressive technique or process called karyotyping. Okay. And so in this method, you can actually isolate cells as they are dividing during mitosis. And you can see here an example of exactly what I'm talking about, karyotyping. And so the staining that actually takes place during this procedure gives us just a remarkable visualization, basically, of the chromosomes as it generates these bands in chromosomes, and you can detect similarities in these bands within the chromosomes. And so based on this practice of visualizing the chromosomes and their banding patterns, scientists allege that 2 chromosomes, which are now, as I pointed out earlier, called 2A and 2B in chimpanzees, fused to form human chromosome 2. Now, to the evolutionists, this accounts for what? Well, it accounts for the disparities in chromosome numbers, where we as humans, they would call, evolutionary taxonomists would call us, Homo sapiens, human great apes. They put us in the great ape family. And they would call the great apes that aren't human, like chimpanzees, bonobos, orangutans, gorillas, they would call them non-human great apes. Okay, so to them, this accounts for the disparities in chromosome numbers, where the other great apes, according to the taxonomists, have 48 chromosomes, we have 46. And these initial images through karyotyping, they're what gave the evolutionary community the initial notion that there may have been a fusion in the human line. But over time, as genetic sequencing technologies began to expand more and more, and technology began to improve, scientists discovered what they believed was basically the setting of this reported or reputed fusion in the human line. And so that's where they found this site in the chromosome 2 spot, where they argue is a highly dense region of these telomeric repeats that should be found if there really was a fusion. But as we can see here, the site itself is actually highly degenerate. And all you have to do is basically look at the forward sequences. You've got TTA, GGG, and then you've got the reverse complement, which is CCC, TAA. Now you can count up the amount of times that you get the TTA, GGG, and you'll notice that there's only a few. There's about 10 instances, and most of them are broken up. They're found in isolation or in independent sections. So your first example of your telomeric repeat is what? TTA, GGG, several sequences in. And then you don't get another one until many sequences in. You get another TTA, GGG. So they're broken. There should be hundreds to thousands of these sequences. This should be a sizable DNA signature. But the fact is, the entire site's only 798 base pairs. It's a tiny signature. And your CCC, TAA, which is the reverse complement, because again, I pointed out DNA is double-stranded. And here you have about 43 instances where you've got a few that are actually head-to-head, CCC, TAA, CCC, TAA. This is very weak. This signature should be a lot larger if this really were the ancient remnants of a fusion. And so you can highlight again, you can highlight the TTA, GGG, these tandem repeats, which you only find in the terms of the TTA, GGG, unbroken once or twice, a couple times at max. In the reverse complement sequences, you only find them 43. You can do this yourself. You can look at it yourself. And what's interesting is the telomere sequences outside of this region completely break down, and they are isolated or separated by hundreds to thousands of bases. And so, Paulogia, use your critical thinking skills. Paul, critical thinking skills are important. Why is this site so degenerate? Why are there only 10 intact TTA, GGG telomere sequences that exist? And why are there only 43 of the reverse complement telomere sequences that are recovered here intact, with the majority of them actually being found in isolation and the sequences outside this region essentially breaking down entirely? It's a pretty pathetic example. And I want to go to the quote from the authors of a prominent paper themselves. And so here you'll notice that the authors of this paper, okay, have a look at this, Genomic Structure and Evolution of the Ancestral Chromosome Fusion Site. They ask, if the fusion occurred within the telomeric repeat arrays less than 6 million years ago, why are the arrays at the fusion site so degenerate? And you'll get your evolutionists like Erica and some of your more informed evolutionists. They'll say, oh, well, this is expected. Why do the creationists harp on this? Why do the creationists always bring this up? Maybe because the evolutionary scientists brought it up first, and they were shocked. They were surprised. This wasn't predicted. This site, again, is only about 70% similar to what a pure fusion site would look like if it were 798 base pairs. And so this site is incredibly small. It's corrupt, but it's also a region that is overlapped by a highly functional gene that we will get into a little bit more later. So going back to the site itself, you can see that even if it's a high density region of these telomeric repeats, which again we find in varying densities throughout the genome, it's circular reasoning to say, well, this is a high density signature of a fusion. No, because now see, that's a relative argument. These critics like Erica that want to say, well, okay, fine. You find other instances of these telomeric repeats, TTAGGG, CCCTAA, in a head-to-head organization in other chromosomes, specifically the chromosome 9, or just in general spread throughout the genome in a non-head-to-head organization, functioning in gene expression, having significant functional activity associated with them. That circular reason, it's a relative argument because it's dense relative to what? Well, if you're going to compare it to, it's subjective. If you're going to compare it to a fusion, no, now it's incredibly corrupt and degenerate. So it's a high dense region of these telomeric repeats that we find in various densities, in all kinds of organizational patterns, in all kinds of compositions, in different chromosomes representing functional DNA elements important to human genes. So it's a high density region of functional DNA elements when compared to other regions similar to this. But the second you want to start subjectively comparing it to a fusion, then no, because it's not pristine, it's weak, it's small, it's corrupt. And at the same time, you don't gain or form highly critical and essential genes like the DDX11L2 gene by slamming together two chromosomes. Okay, this simply doesn't happen. And so, no, the signature is weak. It doesn't work. This is where I want to play Erica's weak video. And so let me just stop share real quick and refresh it, entire screen. So the last sad effort of the evolutionists to basically address this data, and it's been the evolution of the, because you can watch my debate with Erica on the chromosome 2 from over 2 years ago, where she really lacked answers overall to the challenges. But over the last couple of years, she's evolved and said, well, okay, these telomeric sequences are found throughout the genome. Okay, I'll give you that. But it's the only place you find them in a head-to-head organization. And so I want you to see this clip from her most recent video. Paul won't be up to date enough to have any real significant rebuttal to anything I'm saying, which is why he, as a matter of fact, let's check here. It's actually why he doesn't debate the topic of evolution. He just hides behind his scripts, behind the let's see, evolution debates. Let's see how many evolution debates he's had. See if he's had any on the chromosome 2 fusion or the endogenous retroviruses. So no, just all scripted videos, edited videos, repeating arguments from people who we've already debunked, and that's it. Then we get to other people. So Paul, he's scared of the debate dojo. He's scared of the debate octagon. He knows he can't spar with the big boys on these topics. So he's unfortunately not going to be capable of refuting what I'm saying, but somebody like Erica would respond with this, as she has. She puts forth a challenge here, and so let's demolish this challenge. I'm going to go on mute so there's no echo, guys. Just a second.
Just playing around just to see how Donny's work here, his, I guess, recapitulation of Tompkins' work actually pans out. Now, my question is, what the hell was he talking about chromosome 9, about how you can find the fusion sites calling card in chromosome 9? Because as we just saw, you can't. You might be able to find, like, the telomere calling cards, you know, in the telomeres of chromosome 9, but the only place that you find the fusion site in forward and reverse, it's in human chromosome 2. Nowhere else, as we just saw. So double check me, Donny. Go see what you can find.
At the end of the day, this feels a lot like Donny just taking Tompkins' word for things instead of running these tests himself, which, again, he has the tools to do this. I know he does, because, like I've...
So as we know, as is typical of evolutionists, egg all over their face. So Erica now knows, of course, since we've refuted her very thoroughly. The video she was responding to was my open mic impromptu open mic debate with Mark Reed and Grayson from Base Theory. And we found Chris Roop and I did the work ourselves. Dr. Tompkins was not involved. From my understanding, Dr. Tompkins, he was fascinated by discovering this head-to-head organization of the TTA, GGG, CCC, TAA in chromosome 9. Christopher Roop and I did this ourselves. We talked a lot behind the scenes about the arguments put forth by the critics here on the chromosome 2, just like we've done on genetic entropy. You can see our article on that topic here. So if you go to articles, here's our article here, Genetic Entropy, Refuting Arguments Against Genetic Entropy. And so that's a technical article refuting people like Erica, arguments that they employ to basically ignore the reality of genetic entropy. Anyways, so we did the research ourselves. We decimated Erica's challenge. Chris Roop has familiarity, he's got experience running these blast analyses. So he specifically ran the analysis here. And so we found the TTA, GGG, CCC, TAA, and many different chromosomes. Admittedly, they were degenerate. A lot of them were degenerate, but we did find a beautiful signature here in chromosome 9. And it's funny because Erica says, you know, what the heck was Donny talking about chromosome 9? But we've already demonstrated that Erica obviously just took a small soundbite from that video and then ran with it without watching the video in its entirety. Or watching the video, but apparently it was on mute. But also, it couldn't just be on mute. The screen would have to be shut off, essentially, because right on screen, I show this sequence. And so if she was actually paying attention, she would have seen chromosome 9. We actually have a more pristine site than at the chromosome 2. So Paul, tell me, what is so special about the chromosome 2? The density doesn't work. The fact that you find TTA, GGG doesn't work. The fact that you find TTA, GGG, CCC, TAA in a head-to-head organization doesn't work either. Chromosome 9, nobody would say, is a result of a fusion. And notice that it's more pristine than the cherry-picked fusion site. Notice the amount of tandem repeats we get here. Head-to-head. TTA, GGG, TTA, GGG, TTA, GGG, TTA, GGG. You do not get that at the fusion site. Notice the fusion site. They're broken up. They're not found in unbroken chains. They're found isolated outside this region. They totally break down. It's degenerate. It's small. It's pathetic. And then right in a head-to-head fashion, you get CCC, TAA, CCC, TAA, CCCT, and then it starts to break down. This is more of a pristine site than the chromosome 2 site. And yet the chromosome 9, nobody's saying is a result of a fusion, because we're looking at functional DNA elements. We're looking at organizational patterns that are found throughout the genome in all chromosomes, with some chromosomes having unique compositions, unique organizational patterns of functional DNA elements that are important to the health of the cell. This doesn't have anything to do with the fusion. To point to the density of the fusion region might point out something that's unique about this region in the chromosome 2, but then pointing out the pristine nature of, let's say, the chromosome 9 points to a unique feature of chromosome 9. There's unique features and unique compositions found in chromosomes throughout the genome. None of these points make it a fusion, because the second you start saying, well, these DNA repeats are found in high density. Well, high density, what are you comparing it to? It's like when evolutionists say, oh, this universe is old, it looks old. Oh, really? Because that argument's subjective. It's relative. Relative to what? We don't have another universe to compare this universe to. So how do you know that it's old? We don't have another Earth. We don't have another universe to compare it to and say, see, it's old because we're comparing it to this. Yeah, when we compare this to what a pristine fusion site should look like, it's weak, it's pathetic, it's tiny, it's corrupt. So the fusion argument's been absolutely dismantled. Paul, step up to the plate. How do you address this? It's dishonest, it's unsophisticated, it's weak. It's soft to repeat arguments like this, or have a video repeat the arguments for you and not actually address the opposing arguments, the counters, the challenges. Okay? Ignorance may be bliss, but it's not a virtue. And yes, a lot of these evolutionists, they have earned their golden dodgeballs because they have a PhD in the 5 Ds of dodgeball, dodge, duck, dip, dive, and dodge. And clearly that is what Paulogia is doing here. Man, these evolutionists are making it too easy. It's an excellent time to be a creationist, guys. Telomeres, a widespread genetic feature found at the end of chromosomes, have respective calling cards. These are specific sequences found in the genome, and they comprise the DNA letters TTA, GGG in forwards, and CCC, TAA in reverse complement. I want to see one of these evolutionists counter my articles here. Give me a write-up, give me a comprehensive response, put in the work, don't be lazy. These base pairs occur in the specific order at telomere sites. These telomere-specific sequences are found repeatedly at telomere location. So here I go into the fact that we find these interstitial telomeric repeats all throughout the genome that are in spots not specific to telomeres. So defenders of common ancestry, like Paul here, can't look to these sequences found in the so-called fusion spot, the purported fusion spot, and say, see, here's evidence for the fusion, as his video was saying. And not only that, we find these telomere calling cards in both forwards and in reverse elsewhere in the genome, as I've demonstrated here. Okay. Christopher Roop and I worked on this issue to authenticate the prediction. Chris exclusively ran the analysis. He performed comprehensive analyses using this useful program. It turns out that there is compelling evidence for the telomeric repeats, better known as internal telomeric sequences, in both forwards and in reverse being found on multiple chromosomes, but we discovered a similar signature in chromosome 9, the telomeric-like hexamer sequence and its reverse complement are arranged head-to-head in a similar fashion to the purported fusion site. Was the reputed fusion site cherry-picked? It appears so. It is important to note that chromosome 2 is moreover the second largest chromosome in the human genome. Chromosome 9 is much smaller, and yet it contains fewer gaps between the telomeric repeats. We saw in the so-called fusion site how large the gaps were, how degenerate everything was, how these sequences are found in isolation. They're found independently rather in tandem repeat. Okay, and so the evolutionists are quite desperate to look to this so-called fusion site in the chromosome 2 as evidence for some ancient fusion. Not to mention the fact that the 798 base pair region, another reason why it might be more dense when compared to other regions comprising these functional DNA elements, is because the site actually comprises multiple start points for what? For transcription, where RNA polymerase II actually binds. This region is an amazing source of genetic activity, of genomic activity. It is not a fusion site. All the evidence tells us otherwise. And the failure of the evolutionists, the evolutionary scientists, also your content creators on YouTube like Paul, the failure of them to address these challenges, to address these objections, to address these counters, tells us that the chromosome 2 fusion is overturned. It's weak. And it's really, really sad that people like Paul and almost all evolutionists that debate this topic, they look to the chromosome 2 as one of the best lines of evidence. Some say it is the best line of evidence for evolution. We can see how weak it is. It might even be weaker than the argument from endogenous retroviruses, okay? And so, no, it's not representative of a fusion site. It's a functional component inside an essential gene that is completely on fire, and it is highly expressed. It's not what people like Erica say, oh, it's just noise, it's just spurious, nothing really going on here, folks. No, that's easily refuted. They need to drop that argument if they want to be taken seriously, because we're not taking them seriously anymore. Informed creationists like us, we're not taking these arguments seriously anymore, okay? And so this site here is doing a lot. It's active. And the evolutionists, they're really struggling with this one. So notice this. The asserted fusion site is only 798 bases in length, as I touched on. If this really was the mark of an ancient fusion, this region should be much larger than the 798 bases. There are only 10 intact TTA-GGG. Go do it yourself. Telomere sequences that exist at the site, and there are only 43 of the reverse complement. Here's the chromosome 9. Evolutionists, put in the work. Quit just repeating arguments. Quit blindly accepting arguments put forth by other evolutionists like Ken Miller. Look into it yourself. Look into it yourself. And then here's the cherry-picked fusion site. And so, no, it doesn't work. The rescue device doesn't work. Gutzett-Gibbons response doesn't work. What's the fact? The fact is we find the repeats in many chromosomes with a large signature in chromosome 9. It's pristine in both forwards and in reverse. That meets Erica's challenge here. She literally said, what is Donny talking about the chromosome 9? See, she thought I was just talking about, well, you find the TTA-GGG here, you know, independently, and we don't find a head-to-head organization, basically, where you get the TTA-GGG and CCC-TAA, but that is, in fact, what we find. So what's the reality? The suggested fusion site turns out to not be a fusion site. It is a key genomic switch, right? A promoter is a genetic switch within a highly competent gene. The gene that crosses the alleged fusion location is a long non-coding RNA gene. Guys, we now understand that there are many more long non-coding RNA genes than there are protein coding genes. Remember, you have DNA to RNA to protein synthesis, DNA transcribed into RNA, translated into a protein product. But you can have DNA transcribed into RNA where all these RNA products are functioning in a multitude of ways. And the evolutionists, they have a very unsophisticated view of the genome. It's not just the final product that's a protein that is important or essential to the genome. No, it actually turns out that your protein coding genes are more similar across creatures, because the DNA elements that are doing sequences that are doing a lot of the work, the behind the scenes work, are the RNA genes, the non-coding regions of our genome. So that which evolutionists have assumed is junk, representative of evolutionary leftovers, have actually been shown. And we're still just beginning to understand the DNA language, but we are seeing that which the evolutionists have argued are non-functional evolutionary leftovers are actually highly functional DNA elements necessary to sustain healthy life processes in the cell. That includes endogenous retroviruses, allele sequences, pseudogenes. That includes the non-coding areas of our genome. The protein coding genes, they're the building blocks, right? They're the building blocks, which is why they tend to be more similar among creatures. But it's your non-coding RNA genes that are more taxonomically restricted. They're more creature specific. And these are the genes that are actually providing the metadata, you could say, for the building blocks that are the protein coding genes. And so these non-coding RNA genes are actually necessary in order for the protein coding genes to function properly and carry out their jobs. And the DDX11L2 gene is a functional non-coding RNA gene. It is the evolutionists that assume, oh, this is a pseudogene. It was once a protein coding gene. Now it's broken. It was hit with mutations, whatever it is, it's broken. So now it's a pseudogene. It's a non-coding gene. It's basically a leftover remnant of a once fully functioning protein coding gene. That's been completely overturned. Evolutionists that use the pseudogene argument, they're completely out of date on the science, and they're out of touch on reality. It's very similar to a play or a movie where you have the actors and actresses on screen, right? They're like the protein coding genes. But is that it? You just get a bunch of actors and actresses together and then they put a movie together? No. Behind the scenes, which is why your end credits of these big movies sometimes go on for 5 minutes, it seems. That's because you have writers, producers, you have those working in the CGI department, you have those in lighting, you have directors, you've got so many people that are actually you got stunt doubles, you've got so many things going on behind the scenes to make sure that that movie is done right. And that's what we see in the genome too. The genome is a sophisticated network of functional DNA elements. The genome is nested. It's a 4-dimensional code with multiple overlapping codes. It's chock full of meta information. Tell me, Paul, how did this come about? Through naturalistic evolutionary processes. I predict that we'll never get an answer to that question. Consider that a challenge that can't be answered by the evolutionary community. It won't be answered by Paul here. That is a fact. Long non-coding RNA genes, they actually function not just in one way, not just in a secondary way, but in a variety of ways. Many of them function in the nucleus. They are understood to modify DNA. And I've got paper after paper that talk about how these functional RNAs, they actually engage in many biological processes across every single branch of life in the biological world. They affect gene expression. They modify chromatin. And I mean, there's so much going on. I can do an entire lecture on the functional roles of these non-coding RNAs. It also turns out, and I want to go more into the function, but I feel like I am going to get too ahead of myself. And so again, let's go to a little bit more of the video from Paul. And I want to touch on a little bit more of the functional aspects of the region.
Chromosome 2 is really just a fusion of 2 grade 8 chromosomes. Why would you think that? It may be similar to the grade 8 chromosomes because the same designer wrote that code. Microsoft Word and Microsoft PowerPoint share the same spell check dictionary. Probably a million lines of code identical. Does not prove it evolved from explosion in a print shop. Still, it was designed. So similarity in our code or the apes code is because same guy wrote the code.
This line of evidence isn't saying that humans and chimps are related because their DNA is similar. It's saying that we're related because where we are similar, there is a copying and pasting artifact. Again, for the sake of this debate, we aren't trying to determine if
there's no unilateral mistake. There's no copy and paste artifact. Paul, you need to address the points that I've brought forth here. When Ken talks about homology, or we could talk about shared gene order along chromosomes like synteny. Synteny is there for functional purposes. I've got plenty of papers demonstrating that. Functional for genomic architecture. Homology, engineers build homologous patterns. Homology and synteny is not going to be differentiating evidence for evolution. But the fact is, at the so-called fusion site, when you compare it to the chimp chromosomes 2A and 2B, there's actually size differences. There's pieces missing. There's genes surrounding the area in the human genome, in that area, especially around the so-called cryptic centromere site. I'm not sure how many genes, but there's definitely genes there that aren't found in the chimpanzee genomes. There's a lack of ape-specific satellite DNA found around human chromosomes. It's completely missing. And it's found in chimp chromosomes, but it's not in human chromosomes. There's also a massive difference in the telomeric repeats, the interstitial telomeric repeats, when comparing the human genome with the chimpanzee genome. They're almost completely missing. It's totally different in the chimpanzee genome. Paul, tell me how that happened since that split. Where's those changes? So yeah, when Kent talks about homology, or we could even talk about synteny, that would exist for engineering and functional purposes. That would demonstrate common design. But when you actually look at the argument in detail, there's actually a lack of synteny. There's the sophisticated analysis to determine such a thing actually has not been done. Let's continue.
there is a designer, but whether the designer created each kind from scratch, or if the designer built different organisms from common ancestors. If the designer designed humans from scratch, why would the designer add in extra centromeres and telomeres into the chromosome 2 to make it look like?
Paul, you're wrong. You're in error. You need to correct yourself. You don't find the necessary amount of telomeric DNA there if it really was representative of an ancient fusion. You don't find a second deactivated centromere. And what you actually see there, that evolution is trying to say, see, this is, alphoid repeats indicative of a deactivated or second centromere. That's actually overlapped by a functional gene. And so you're just wrong. We don't find that. When you see those chromosomes come together, and there should be this massive signature, thousands of bases long. Again, what we're looking at is 798 base pairs. It's weak. And the reasons that the authors of these prominent papers, one specific on chromosome 2, give for why it's so degenerate, they don't work. Paul, you're repeating false arguments.
Like gene fusion, when we would expect that perfect designer to instead implement a clean, from-scratch copy of his design.
How do you reconcile the chromosome 2 fusion in humans?
He's saying there are similarities. We're getting rough on a different topic, but there are similarities in the chromosomes of humans and chimps, he thinks. Actually, people don't, you've got to really study that. Okay, I could put documents and say, look at the similar documents. These two documents both use the same 26 letters of the English alphabet. That proves they're related.
Again, Kent misses the whole point. For the sake of this debate, I'm agreeing that there's a common designer. We're not examining the similarities or differences of chimps here. We're asking why the designer would use a clean copy of the code for chimps and an obviously mangled copy of the code for humans. It's the centromeres and telomeres that need to be explained in Kent's fixed.
Again, the necessary telomeric DNA that should be found at the site. If it really is representative of an ancient fusion, it's not found. We don't find the cryptic centromere. And so it's not obvious like Paul wants to say. And so you can really see how unimpressive, how unsophisticated his video is here, because he's not interacting with any of our actual arguments against the chromosome 2. He's not putting in any of his own work. It's the same thing with endogenous retroviruses. It's even worse with ERVs. He relies on others, like stated clearly, whose arguments have already been demolished. We just hit the hour mark. And so, ladies and gentlemen, I am going to take just a 30-second break, and I'll be back to continue refuting Paulogia. All right, we're back, and let's continue this chromosome 2 fusion beatdown. I want to go over a few slides and demonstrate more as to why Paulogia has no idea what he's talking about. Okay, so here we go. We've got the so-called fusion site, the very, very weak signature, huge gaps. It's very degenerate. And when he keeps saying, oh, Kent specifically in the video, but we got to explain the telomeric DNA that's found. We got to explain the signatures of a fusion that are found. The signatures aren't there. Paulogia, it's not there. And the fusion site itself is actually read in the reverse complement. It is a transcription factor binding site, again, in the middle of a long non-coding RNA helicase gene. It codes for a functional RNA molecule that is functioning in the genome. And so for the third or fourth time, Paul, how do you get highly functional and essential genes like this long non-coding RNA helicase gene by slamming together two chromosomes. The site itself is not representative of an ancient fusion. It is itself an essential genetic switch, which is called a promoter, that controls and regulates the what? The gene that overlaps the area, the DDX11L2 gene. It also binds to 12 different types of transcription factors, okay? Transcription factors, guys, they are proteins that basically latch onto a gene and turn it off and on, or tell it how fast to run and how much product to make. There is so much control. There is so much sophistication going on in the genome. The genetic systems that make up life, they're elegant, they're complex. They could not have come about through evolutionary processes like natural selection and deleterious mutation accumulation. RNA polymerase II also binds, as I explained earlier, inside the fusion site and starts transcription. Paul, how do you explain all of this? You can't. What might this DDX11L2 gene be doing? Notice this. It's expressed in at least 255 different cell or tissue types. It's also co-expressed, turned on at the same time is what that means, with a variety of other genes. And it's connected to processes associated with cell signaling in the extracellular matrix and blood cell production. So when these evolutionists like Erica want to say, oh, it's a pseudogene, the activity associated with it, it's weak, it's spurious, it's just noise, they don't know what they're talking about. They're desperate to hold on to this argument because Ken Miller said if there was no fusion, there's no ancestry. And they desperately want to be related to a chimpanzee. It was Erica who said, when I'm at the zoo and I'm locking eyes with the chimpanzee at the zoo. It's like I'm looking at myself. It's like I'm looking at a reflection of myself. I mean, it's sad how badly they want to believe they're related to walking sticks, strawberries, and whales. And so they have to invoke a lot of pseudoscientific logic, pseudoscientific related arguments like we've seen here. Not only that, the DDX11L2 gene, it's actually situated in a region within the genome with many other important genes, many other significant genes. And so it exists within a gene-rich area of the genome. We're seeing a lot of activity, those telomeric sequences again, TTAGGG, CCCTAA. They are found all throughout the genome in all chromosomes, and instances where they're in a head-to-head organization. There's nothing special about the so-called fusion site, and the dense nature of it doesn't work. It's relative, it's subjective, it's weak. The evolutionists, they have to argue, they have to drop this, essentially. And then what they'll say is, oh, well, this is, Dr. Jeffrey Tompkins, who has done fantastic work on the topic. They'll say, oh, but, he's a creationist. So, they don't accept that. He's a creationist, which is just really weak. It's really weak. It's because they can't refute the data. And so they have to say, well, he's a creationist, so we can't trust him. No, the data is sound. The data is strong. It's undeniable. But for those evolutionists that just, can't seem to accept that, and they still want to continue repeating these weak arguments that the DDX11L2 is not actually functional, well, you know what? Just check the genetic database yourself, which I've done here for you. Go verify all of this. You don't have to take my word for it. You don't have to take Jeff Tompkins' word for it. Simply search the DDX11L2 gene, and you'll find that the DDX11L2 gene has almost 2,000 functional associations with biological entities spanning 7 categories, extracted from 20 data sets. See, here's the problem, is the evolutionists, they assume that the genomes of biological organisms, they've been built up through inadequacies. They've been built up through mutations, natural selection, environment. And so they don't look to the genome to find treasure like creationists do. And so they don't understand that genes are now understood to be associated in sophisticated networks that interact with each other. And therefore, you need to look for what's called co-expression. when looking at the functionality of these non-coding RNA genes. And so when creationists that are up to date on the literature that understand this, like Dr. Jeff Tompkins, PhD geneticist, when they actually look into studying these genes the right way, we see just how functional they are. And the evolutionists can't deal with it because they're desperate to hold on to this argument. And you heard Paul, he said this was definitive, even though he had none of this that I'm saying tonight, for the most part, had he known about. So you'll see here, you can do this through BLAST. Those telomeric repeats, right? You can see them in chromosome 2. You can see them in the middle here, and you can see color code, how much are found. Well, notice you found them on 16. You find them in 22, 21. Here's chromosome 9, where we find them in a head-to-head organization. You find them in many chromosomes, and you find them scattered throughout tiny chromosomes too. Look at 22 and 21. Way smaller than chromosome 2, and yet you find quite a bit. They seem to be present in high density. But again, they'll say, oh, well, most of those areas, they're not found in mirror form. They're not found TTA, GGG, CCC, TAA in a head-to-head organization. Oh, well, we do find that in many chromosomes, but we find a strong signature in chromosome 9, as I've demonstrated. Notice this. Human chromosomes 2, 16, 21, and 22 were peppered with the so-called fusion site sequence over the length of the entire euchromatic landscape. You know what Erica does is completely circular. So she takes the fusion site, basically. She takes this big site, and then she searches throughout the genome using BLAST to see where that site's found. Obviously, it's going to be found in the chromosome 2. That's its composition. And then she says, see, it's not found anywhere else. And so, the only place you find it is in chromosome 2. That's what you're looking for, though. Go take the chromosome 9 sequence that's quite pristine. Go search that in the genome. It's circular reasoning what Erica has done. But I don't think it was Erica that first did it. I think it was her evolutionary biologist friend, Evograd. And so it's circular what they did in the first place. That's probably why they didn't find what we found. That's why it went completely missed. But now they want to look to the density nature of it, which is relative. It's subjective. They're going to need something a lot better than that. And so, guys, the evidence is clear. This so-called fusion site, we now understand, is an active promoter or a genetic switch. It comprises numerous start points for transcription, where, again, RNA polymerase II binds. There's also multiple transcription start points within this region. The site is a hotspot for genetic activity. And the evolutionists that want to continue saying, oh, but this is the ancient remnant of a fusion, the mark of a fusion. No, because healthy telomeres in humans and other mammals, they're composed of the TTA GGG, right? The 6-base repeat for thousands of bases in length. And healthy telomeres in humans typically consist of 800 to 2,500 of these TTA GGG repeats in complete tandem. But what did we see in the so-called fusion site? Let's go back to it. Huge gaps, incomplete, mostly broken. We don't find many of them in tandem repeat. And so even if we were to imagine, because evolutionists love to hope, dream, and imagine that they're related to fish flies. No, the data says otherwise. Erica, you're not related to a mosquito. Okay. It's not what the evidence. Now you can believe you're related to a mosquito, but it's not science. You can believe you're related to a chimpanzee, but you can't use the chromosome 2 fusion argument because it doesn't work. It's been overturned. It's been falsified. Okay. So even if you were to take 2 of the tiniest size telomeres and fuse them together, we should find a DNA signature or sequence of about 10,000 bases in length. You can even say it's degenerated significantly over time, and you'd still be looking at something that's 3,000, 4,000. Give them 2,000. No, this is 798 base pairs, and we're supposed to believe that this is a leftover fusion vestige, basically. And then the larger the telomeres are that actually fuse, because for sake of argument, we're just saying take the two tiniest, the larger the signature should be. And then the worse it is for the evolutionary narrative. We don't find a signature anywhere in this range. And so Paul's video here, it's very weak. And unfortunately, his audience will just watch this video. They'll watch his video. They'll eat it up. They'll blindly believe what he's saying is true. And arguing that, most of the so-called signature has been lost over time, it's assuming that it's a fusion in the first place. No, this is an obstacle that they can't overcome. Paul can't. So Paul wants to say the creationists have to address the signature we find, right? You saw him say it. But then we show him that the signature is not the signature that we should find. He said, well, most of it's been lost or degenerated over time. See how they want their cake and eat it too? Well, is the signature there or is the signature not there? Pick one. Because when we actually look at this so-called site, there is a noticeable lack of unbroken telomeric repeats. There's a lack of them. And then the intact ones, as you can see, they're primarily found independently or in isolation. And why that is because the other bases, they don't exist as tandem repeats where you have TTA-GGG, 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 like we find in the chromosome 9 or at the end of chromosomes. So no, this is not the ancient relics or leftovers of a fusion, because we should locate thousands of complete motifs in near perfect tandem, and we don't find them. And again, the concentration of telomere sequences completely breaks down outside of this region. And Paul's never addressed any of this. Here we go. Long non-coding RNAs that I was talking about earlier, backing up what I'm saying. Long non-coding RNAs are a diverse class of RNAs that engage in what? Numerous biological processes across every branch of life. Gene expression patterns have implicated these types of RNAs, long non-coding RNAs, in diverse biological processes, including cell cycle regulation, immune surveillance, and embryonic stem cell pluripotency. Go look up the gene yourselves, guys. I can't do all the work for you. You're going to have to do it yourself, because this whole pseudogene argument saying that the area is not actually functional, it's just noise, it doesn't work. So let's see how much more of this video we actually have. I wonder if Paul ever actually makes a difficult argument. I don't think so. Let's see.
Kind's view. He took one last crack at this because Kant loves to default to complexity.
God made human chromosome number 2 to let us know evolution did happen. If you have to think of it in those terms. I don't have to think of it in those terms, and I don't intend to think of it in those terms.
That is why you fail.
Human chromosome number 2 is incredibly complex. Any one chromosome in any cell is more complex than the space shuttle, the most complex machine built by man, as far as I'm aware. Okay, one chromosome is more complicated than the space shuttle, and if you think that is proof that God made it happen by chance, you need serious help.
Again, Kent, no one discussing human chromosome 2 is denying that it is complex. Just the opposite. We're saying that it seems to be needlessly complex. The designer mashed two existing chromosomes together rather than designing a single chromosome. The best answer for this is that the designer utilized common descent. Finding Tiktaalik. As you'll recall, what science values most.
Notice how disappointing. That's it. Guys, that is it. That is the definitive. No wonder why Paul was unsurprisingly absent in our evolution debate challenge, which we did about 60 of them. I personally did several on endogenous retroviruses. Where was Paul? Nowhere to be found, because he doesn't want to enter the debate dojo on these topics, or the debate octagon on these topics, because he can't deal with the counters to his weak arguments. Those arguments deserve to be put in the trash. I'm sorry if that sounds harsh, but here on Standing for Truth, we don't believe in soft apologetics. Soft apologetics, they don't work on these kinds of militant evolutionists, okay? And so when Kent says that the human chromosome 2 is highly complex, He's right. It's functional. And Paul keeps saying, you have to explain why the area looks like it's an area that's been mashed together. No, it doesn't. I've already demonstrated that it looks nothing like that. It's a weak signature. It can't be compared to a fusion site because it's too small. And what we find here, the type of signature and the organization of these sequences are found elsewhere. And when Kent says to Paul that chromosome 2 is complex, yes, the fusion site, which isn't a fusion site, comprises multiple start points for transcription where RNA polymerase 2 binds. The fusion sites overlap by a highly functional gene. And this video is very unimpressive. Okay, let's go through a couple rebuttals to their spurious argument about the so-called DDX11L2 gene. Okay. Strongest evidence against a fusion. The purported fusion site read in the minus strand orientation is a functional DNA binding domain inside the first intron of the DDX11L2 non-coding RNA helicase gene, acting as a second promoter. The fusion sequence binds to at least 12 different transcription factors. I talked about all of this. The DDX11L2 long-known non-coding RNA gene containing the alleged fusion sequence encodes several transcript variants and expressed, and notice this, in at least 255 different cell and or tissue types. The fusion site itself appears to be the function. So then the evolutionists say they don't even know what the fusion site is doing. It appears to be the functional start site for the shorter transcript variants. Furthermore, annotated DDX11L2 gene transcripts suggest complex post-transcriptional regulation through a variety of microRNA binding sites. When I had my impromptu debate with Dr. Dodgeball Dan Stern-Cardinal, who couldn't get out of there fast enough, he knew he couldn't spar in the debate dojo on this topic because it's a topic that's indefensible for the evolutionists, at least when they're going up against the creationists who studied it and can counter their arguments, because their arguments are a bunch of nonsense. And so he kept saying, oh, well, you know, RNA polymerase 2 just likes to bind to things, and the genome's really messy, and the DDX11L2 is spurious in its noise, and heterochromatin, right, this densely packed pieces of DNA basically that mainly comprise repetitive sequences. That's just junk. It can change over time quickly because it can be hit with mutation. Selection's not going to preserve it or purify it. Essentially, there's no purifying selection, he'll say, on areas like that. And just all of these arguments demonstrating that he is starting with an assumption that evolution is true. So prove evolution by assuming evolution, and assuming that the vast majority of DNA, including, you know, DNA elements, DNA sequences, heterochromatin, so-called pseudogenes, are just reflective of evolutionary leftovers, genomic fossils, and a long history of mutations. But that's not what we find. What we find here is activity. This is function. There's no way around it. Paul, let's see you get around this one. Many of these microRNA binding sites are shared with the DDX11 protein coding gene transcripts. In fact, both the DDX11L2 and DDX11 genes are significantly co-expressed. Guys, we have co-expression here together in the same tissues. Shared microRNA binding domains and co-expression putatively suggest co-regulation between a protein coding gene and its non-coding RNA pseudogene counterpart, as depicted in the classic case of PTEN and PTEN pseudogene transcription. And so it's not looking good for the evolutionary community. It really isn't. And so I think we're going to basically wrap it up there at roughly the hour and a half mark. But I do want to encourage people to go over to my article here, Overturning the Human Chromosome 2 Fusion. Paul, put in the work. Let's see a detailed rebuttal, point by point, to my article here. And then try and save the day for Erica, because she's struggling. See the reality here? The reality is Gutsy Gibbons struggling. She put out a challenge. The challenge has been met. And It's funny to see the evolution of Gutsick Gibbons rescue devices over time. Again, go see my debate with her from a couple years ago. She wasn't saying the same thing she's saying now, because what evolutionists do is they go back to the drawing board. They get refuted in debate, and then they go back to the drawing board, and they say, instead of tapping out, they say, oh man, how are we going to save the day for the fusion argument? And so they come up with more and more rescue devices. The objective evolutionists would actually tap out and say, you know what? Ken Miller said without the fusion, there's no common ancestry. And so there's no common ancestry. They should recognize the gene, the DDX11L2 gene. It functions in over 200 different cell types. And the so-called fusion site is overlapped by this gene. You're not going to get highly functional genes by slamming together two chromosomes. The gene itself is linked, as we just read, to the expression of other genes. And these are genes that are at the top of the pyramid of so many important cellular processes. Come on, Paul. Let's see what you got. Because what we saw in that video, it was weak. It was soft. It was unsophisticated. It was unimpressive. And Paul, you got to do better. And the ERV section is no better. It's also weak. It's also soft. It's also unsophisticated. And it's also unimpressive. And I am going to have a blast demolishing Paulogia on endogenous retroviruses. So with that, guys, share this content around. The truth is so incredibly important. Even your most militant critics and your most militant content creators, this is the best they have to offer. They can't address the best argument for separate ancestry. And that's why I always say it's a great time to be a biblical creationist. God bless all.